This slide's discussing the processing and the assembly and stabilization of the class 1 MHC molecule within the rough endoplasmic reticulum. You have the ERP57, and then you have the class 1 alpha chain. Remember, it's made up of three pieces. So this third domain is kind of sticking out there. And imagine this is a two-legged man with one leg cut off. It can't really function very well. And it's in order to open the groove up and allow things to come together, there's a series of events that have to occur. So calnexin is one of the first kind of accessory proteins to come into play. It binds along the alpha one region of the class 1 MHC. And once that happens, then you can have the ERP57 can bind and form this complete complex. Once that occurs, then it kind of opens it up and makes room for beta 2 microglobulin to knock the calnexin off, which will allow the capsaicin and calreticulin to bind and associate with the surface of the complex. That tends to migrate toward the area where your TAP proteins are associated. So your TAP proteins are going to be bringing in peptides. And when they come in, those peptides can bind into this open region. So they've kind of got that, the capsaicin and the calreticulin are kind of opening up that box area that's the groove for the antigen processing and binding. ERAP is an enzyme that helps to transport and bring that in. It's just an accessory protein. And then once it binds into place, then you get disassociation of all these accessory proteins. And it moves into a different, I think it moves into a lipid raft that will then target it toward the Golgi apparatus. Once it gets to the Golgi apparatus, then it's sent to the surface of the cell. So let's look at this. This is how antigenic peptides are generated using the exogenous pathway. And then you also get recycling of receptors that are occurring here. You have to remember that in phagocytosis, you can have the FC region of antigens, of antibodies binding to the surface of the cell. And if they bind to the antigen, then they can trigger clathrin-coated vesicles. Usually it takes more than one to trigger vesicle formation. And it brings it into the cell. Once it's brought into the cell, then hydrogen ion pumps start pumping hydrogen ions and altering the pH, which cause disassociation of the antigens that have been processed from the antibody that's out there. And in this early endosome, that pH is going to be around 6.0 to 6.5. Well, once that's done, then they can separate out the peptides for processing and recycle those receptors to go out and do more work of what they're supposed to do. Once you have these antigens, they can bind into the MHC class 2 and be expressed directly, or they can go into some of these late endosomes. So what can happen is in the rough endoplasmic reticulum, the MHC class 2 molecules are linked with this yellow Li protein that's associated there. And it has a little, I guess you would say, plug that holds that groove in the right place. And what can happen is in the late endosomes, when the pH continues to get more and more acidic, you get 4.5 to 5.0, that cleaves some of that leg that's off of there, and it allows the reassociation of the antigen pieces into the groove, which then can come out. So every once in a while, you'll have some antigens that are the right size piece to fit into the MHC class 2 molecules. But in many cases, you're going to have these dash LIs that have to process the antigens a little more due to the differences in the pH causing association of those things. So that gives you a brief overview of the recycling. Invariant Li chain, it leaves a clip Then peptides are processed, and it releases the clip because of the pH, and it inserts into the region. So you can have different heterozygous types of type 2. Remember, there's 8 of them in a um, typical organism, and you can see that there are differences, and some of them will react to the peptides and some won't, and then you'll get this clip there. So what it's trying to do is maximize the opportunity for those peptides to be expressed in the MHC class 2 molecules. And here is, I talked about the exogenous pathway before, uh, and here is a slide that just walks you through the steps. I don't think I need to sit here and read this to you. You are college students. You should be able to read through this particular slide. And in fact, I would recommend you spend a little time looking at this slide, making sure that you can draw